A warm welcome to all my dear friends. Welcome to Unacademy Offline Center, Kochi. I'm Arun R. Nair, your botany faculty. Today we are going to talk about a topic that is called gene regulation and Lac Operon. And this is very important for your NEET examination. Believe me, every year one or two questions definitely they ask. That's why we have chosen this topic for you, and stay tuned with us. We will be covering this topic very beautifully. Okay? So first of all, we'll understand what do you mean by gene regulation and why we have to do this. Fine? So in our body, we have what? Our genetic material, which controls our all the things. So if you look at a cell, so this is your cell, which have a organelle called your nucleus. And this nucleus consists of what? The genetic material that we say as DNA. So DNA is the genetic material which controls all the regulations that happen in our body. So it is going to make what? We have studied the central dogma that DNA will make. RNA. RNA will make what then further? Protein. So this is what we are talking about, DNA to RNA, RNA to protein. So this is what, this is we call as central dogma. We have to study this. So DNA to RNA, this process is called as what? Transcription. RNA to protein is called as what? Translation. Got it? So these all topics we have previously covered. Now we are talking about here is what most important is the gene regulation. So in our body, there are a lot of proteins are made. Some proteins are required, some proteins are not required. So on the basis of the metabolism, whenever the proteins are required, we want to synthesize those enzymes. Whenever the proteins which is not required, we don't have to synthesize. So who is going to decide that? That's what the regulation will come, gene regulation. Constitutive. Another is called non-constitutive. What do you mean by constitutive gene? Constitutive genes are those genes who we never regulate. Why we need to regulate? Because every time their enzymes are required. I will tell you one example, and this they ask also in the NEET, like for example, glycolysis. Glycolysis means your sugar splitting, glycose plus lysis, sugar splitting. So in this glycolysis, there are certain enzymes which are required, and every time we know that in a cellular respiration, the glycolysis enzymes are required. So we never regulate these enzymes. Every time these enzymes are what they are continuously produced. Such genes are called constitutive gene or housekeeping genes. But on the other hand, there are certain enzymes which only at the time of what metabolism is required, and those genes are called non-constitutive genes. And the regulation that we are talking about is actually we are doing on this non-constitutive genes. Got it? And this is also known as luxury gene, smart gene, all this. Got it? So to understand this constitutive gene, we have one system called Lac Operon. With the example of Lac Operon, we will try to understand how we can do this regulation. Got it? So we are talking about gene regulation. So which are the places where we can do the gene regulation? So it depends on the type of cell. If you talk about eukaryotic cell, eukaryotic cell. So we have got 4 places where actually we can do the regulation. And what are the places? First, we can do this at the transcriptional level, where the formation of primary transcript takes place. Second, we can do at the time of splicing, where the processing we do for what? We do processing for our splicing to make the RNA stable, and also we remove the introns, those non-coding part. Then third place where we can do when we want to transport the mRNA from nucleus to the cytoplasm. So when we are transporting mRNA from nucleus to cytoplasm. At this place also we can do the regulation. And the last where we can do is at the translational, where we are converting the RNA to your amino acid. Converting means basically information will be there, those information will be read, and on that basis a particular sequence of amino acids will be joined together, and thus we get the polypeptide chain. So that is the translation. So these are the 4 places where in eukaryotic cell we can do the gene regulation. Same thing if I want to see in prokaryotes, prokaryotic cell. This we mainly do at only one place, that is at the transcriptional initiation. Transcriptional initiation. So this is the main place where we are focusing in the prokaryotic cell. Got it? And this is what we are going to study more detailedly. To understand this, we have got what? One Lac Operon model. Lac Operon. What do you mean by Operon? Operon, we can say that in 1961, 2 French biochemists, Jacob and Monod, those 2 people were actually the one who actually come up with this concept called the Operon model. They have observed in the bacteria that there are group of genes are there that we keep it together. So if you look this, so here we can see that there is a group of genes are there which are placed together, and those actually called as what? Operon model. So what we keep under this? We have 4 genes. One we can see here, it is the promoter. Another that we can see is the regulatory gene. Another that we can see is the operator. And the last one that we see here is the structural gene. So we can clearly see that these are the places where this is what the structural gene we're talking about. These are these regulatory sequences. We call this as what? Promoter. This as operator. This we have that regulatory gene, and this is the same promoter. Got it? So what we are saying that Lac Operon, particularly the lac means, here means what? Lac, that is lactose. So this gene particularly regulate the metabolism of what? Lactose. Now this is what we have to understand more detailedly, and in your NEET examination also, so many questions they ask from here. So this is a very, very important topic we have to understand. Okay, so we'll start with the very basic, we'll understand how this entire Operon model is going to work. Fine. So what I said, they have got what? Operon means, Operon is a combination of all 4 these genes. They have promoter gene, regulatory gene, operator gene, structural gene. So we can see that this is the combination. This is a perfect example of what? Lac Operon. Lac means lactose metabolism, Lac Operon. So here we can see this particular combination of genes will do the metabolism of your lactose. So here we can see promoter gene is there. Here we can see the operator gene is there. We can also see the structural gene, ZYA. If you look very closely, we are talking about what? This happened in a prokaryotic cell. So in prokaryotic cell, this is what you're looking. You're looking a polycystonic condition. We'll discuss that also, fine. Then we have what? A regulatory gene, this one. Got it? So this is what we're going to talk a little bit more detailedly. So like I said, now we are going to detailedly talk about the Lac Operon. So this particular Operon model that we are talking about is in the bacteria. So we can take this bacteria, that is your Escherichia coli. Okay. So this is the bacteria, they can see peritrichous condition. So this is your E. coli bacteria. So in the E. coli bacteria, they have this particular gene combination that is called Lac Operon. Similarly, many operons are there. Okay, not only Lac Operon is there, Trip Operon is also there. In your NCRT it is given, histidine is given, valine is given, ARA, okay, arabinose also given. So all those operon models are exist in our bacteria. But these particular operon model they have studied first, and this is the most important one. And in your NEET also every time they ask from here only. That's why we are focusing more towards the Lac Operon. So what happened? Very simple logic. Just imagine this bacteria is feeding on what? Is feeding on the glucose. We know that every organisms, their first preference will be always be what? It will be carbohydrates, because that's the easy way to absorb. So if this E. coli is feeding on lactose, so do you think that it needs to switch on this particular gene, structural gene, who actually do the metabolism of lactose? Just imagine if this gene is on state, that means this structural gene, from this gene they are going to make the RNA, and from that RNA they will make the proteins, and that protein is enzyme. So just imagine those enzymes are necessary for what? They are the necessary for the metabolism of lactose. So now the bacteria is feeding on glucose. So do you think they need to switch on this gene unnecessarily? Unnecessarily they will spend a lot of energy, because we studied in transcription, translation, lot of energy is required to switch on this. So that means they put a lot of energy, they make the enzyme, and after making that enzyme, there is no use of it. So why the bacteria want that? So that means bacteria do not want that lactose enzymes to be produced unnecessarily. So it will be in which state? You are saying that it will be in a switched off state. It will be in a right now in what state? Switched off state. So like I said, this particular operon which we are talking about, this particular gene is right now in a switched off state. So what we want, when you cut down the supply of glucose and when it will start to feed on lactose, at that time we require the enzymes, because those enzymes will do the metabolism of lactose. So we'll understand this all entire thing, how it's going to happen. So like I said, right now if lactose is not there, at that time glucose is available. So what is going to happen here? The regulatory gene which you see right now, it is a type of a constitutive gene. Constitutive gene means it always in a switch on condition. So it will always make its respective RNA. DNA to RNA, that is called transcription. Transcription. And this is which type of RNA they will make? RNA which is called repressor. So this is a repressor RNA. And from this repressor RNA, they are going to make what? They are going to make a protein, which is enzyme in nature, or we can say basically a protein. So this protein that is they make, that is called repressor protein, which is a type of an active repressor. Now this active repressor has a binding efficiency, where it will bind. It can go and bind with the operator, or it can also bind with the lactose also. But right now, do you think the lactose is there in the system? No, there is no lactose. It's feeding, bacteria is feeding on what? Glucose. So since there is no lactose, this active repressor which is formed, it will go where? It will go and bind over the operator. Now what? It will make a difference. See, we know that how they are going to do the transcription. Always you see in the promoter who will come? The RNA polymerase. So you can see this is your RNA polymerase. So always RNA polymerase will come in the operator side, it bind and then it do the transcription of this DNA to RNA. That process is called transcription. But right now, when RNA polymerase will come and bind with the operator, you can see with the promoter, who is in the operator side? Your active repressor. Because of that, RNA polymerase cannot do the transcription, and since there is no transcription, the gene will be in which state? Switched off. And it's logical. So you see, when bacteria is feeding on the glucose, okay, in our intestine also E. coli is there. So when you are just feeding on the carbohydrates, glucose, easily when they are feeding on the glucose. So why they have to unnecessarily make the enzymes who would do the metabolism of lactose? Now just imagine, now glucose is not available. Now what is there? you started to give the lactose. Now lactose will come into the system. So this is going to be a constitutive gene. So from your regulatory gene, I does not stand for inducer, it is an inhibitor. Okay, in NCERT also it is mentioned. From here also they ask one question like what is I stand for? So we know that I stands for what? It's a inhibitor, not an inducer. So this regulatory gene will again make the repressor RNA. From this repressor RNA, they will again make the active repressor. But this time, we know that when you are giving the lactose, lactose will come inside. How lactose will come inside? There will be a little bit amount of what? Enzyme that is called a permease enzyme will be there. And that permease enzyme will allow the movement of lactose. Once the lactose will come inside, it can be in a form of what? It can take the form of your allolactose, which will act like a inducer. What do you mean by inducer? You'll understand. Inducer is the substance, any chemical substance that will make this active repressor into inactive, and because of that, this gene will be changed from switch off to switch on. Right now it is switch off, now you have to make it switch on. For that, if any chemical substance which make the gene from switch off to switch on, so that substance is called as what? Inducer. In this Lac Operon, question will be asked, which is the inducer here? So answer will be allolactose or lactose also, you can say. In NCERT it is written, so lactose or allolactose. So here the inducer, when you give, glucose is not there. It's a one of the condition. When just imagine, glucose is not there, lactose is available. So it has to get energy. So it will do what? It will now uptake the lactose. A little bit permease will allow the movement of the lactose inside. Lactose will come inside, its allolactose form will bind with the active repressor. You can see it is going to bind with this active repressor. What it will make the difference? Now, no longer it is able to bind with the operator. Operator is free. You can see operator is free because this active repressor, now it will become what? It will become inactive repressor. Why it become inactive repressor? Because active repressor is continuously forming. Now you have given the lactose. Lactose will bind with what? With the active repressor, make it as what? Inactive repressor. Got it? So now it is not able to bind with the operator. Now this RNA polymerase can move. Yes, it can move. So when it will move, the polycystonic condition, we have one gene Z, Lac Z we say, Lac Y, Lac A. They are going to make respective RNA. Okay, this is the RNA. Now from this RNA, they will make the enzyme. What enzyme they will going to make? From Z, very important question will be asked. Beta galactosidase. This enzyme will be do. They will make. What this enzyme is going to do? We will understand. From Y, they will make permease. A, they will make trans acetylase. Now what are their functions? We said that when, again I am repeating, listen, when regulatory gene will make the RNA repressor, that is from that RNA repressor they will make the active repressor. Now lactose is available, no glucose, so lactose will act, allolactose will bind with the active repressor, make it inactive, so no longer this is able to bind with the operator. So now what will happen? RNA polymerase is able to do the transcription. So now gene is in which state? It is in a switch on state. You have regulated the gene. Now this is in a switch on state. Now from this gene, that means this DNA to RNA, from this RNA to your protein. That is, they make the enzymes from from Z. They will make the beta galactosidase. From Y, they will make the permease. From A, it is going to make trans acetylase. We understood the function of permease. It will do what? It will do the allowment. It will allow more lactose. So more quantity form. So more lactose will come. Now when more lactose will come, what will happen? Now they are ready with the enzyme. What is this function? Enzyme is going to do? It is going to break. What is lactose? Lactose is a combination of your glucose plus your galactose. So this enzyme which we talked about, which one? Beta galactosidase. Beta galactosidase. It is going to break this into its monomeric form. It's a disaccharide. Who? Lactose. Lactose is disaccharide. So now when lactose will come, Now they are ready with the enzyme for the metabolism. That beta galactosidase will break the lactose into glucose and galactose. Now you can derive the energy from the glucose. Thus, you made the gene from switch off to switch on. That's what the regulation we did. So Lac Operon follow which type of system? We can say it's a called inducible system. Lac Operon follow what? Inducible system. What do you mean by inducible system? Whenever gene is already in a switch off state, from switch off to you make what? Switch on. For that you use a chemical substance. That is called as what? Inducer. So from switch off to switch on, when you do that type of system is called inducible. Can we see that here? Yes. Initially the gene was in a switch off state. Why it was a switch off state? Because from the regulatory gene they are making the repressor RNA. From that active repressor it is going to bind, it's going to bind with the operator. RNA polymerase is not going to do the transcription. Gene was in a switch off state. Now what happened? Bacteria started to feed on lactose. Glucose was not there. Lactose came. Who allowed the movement? Permease. A little bit permease will be there. Permease allowed the movement of lactose. Now lactose, when it come, allolactose form. Inducer bind with the active repressor, make it inactive. No longer able to bind with the operator. What happened? RNA polymerase will able to do what? The transcription. Gene become from switch off to now switch on. Logical. Why? Because now when more lactose will come, we have to do the metabolism of lactose. We have to break the lactose into glucose and galactose. For that, what we want? We want beta galactosidase enzyme. And do we have now? Yes, because gene is switch on. So that's why we have beta gal. Question will be asked in NEET. What Z will make? Beta galactosidase. What Y will make? Permease. What transacetylase will do? Transacetyl will do. It also have a function. It add the acetyl group to beta galactoside. Got it? So in this way, you can see gene become from switch off to switch on. Similarly, if lactose is not available, just imagine lactose is not there, again they start to feed on glucose. When lactose is not there, so do we need these enzymes to produce? No. So what happened? There will be no more allolactose. This will become an active repressor. It will go and bind with the operator again, and again RNA polymerase will not able to do the transcription, and thus it again become what? Switch off. So in this way, we do the gene regulation in this particular prokaryotic cell, which is we're talking about your bacteria, where we do this. Very important. We can say that every year they ask this, their favorite, NEET favorite, they ask one question from here, 1 or 2 questions, their favorite. They can ask, this is the area where they will focus like inducer, they focus on this enzyme, what this enzyme do, from which gene, what is their enzymes is produced. It's very, very important. So this is your very small topic that we covered today. That is your gene regulation and Lac Operon. And further, if you want to learn more from our Unacademy offline center Kochi, please come and join us. Thank you so much.